And welcome back to another video where today we're going to be discussing enzymes, factors that affect enzymes and their rates, and enzyme inhibition. Okay, so let's dive straight in. Enzymes are biological catalysts, meaning they speed up chemical reactions without being consumed in the process. One of their key roles is to lower the activation energy required for a reaction to take place. This allows reactions that are vital for life to occur much faster than they would on their own. Without enzymes, many processes would be far too slow to sustain life. Optimal conditions are crucial for enzymes to function properly. In the human body, enzymes are most efficient at around 37 degrees Celsius, which is relatively low compared to industrial chemical reactions. This enables metabolic processes to occur at normal body temperature without needing excessive heat. Structurally, enzymes are globular proteins. Their 3D shape is critical for their function because the active site, where the substrate binds, depends on this specific structure. If the structure is altered, for instance by changes in temperature or pH, as we'll discuss later, the enzyme might lose its functionality. When a reaction occurs, the enzyme binds to the substrate at its active site, forming what's known as an enzyme-substrate complex. This complex is essential because the formation of it is what facilitates the chemical reaction. Now, a key concept in enzyme activity is the induced fit model. In this model, the enzyme's active site is not an exact fit for the substrate right away. Instead, it molds around the substrate as they bind, much like a glove fitting a hand. This slight change in shape helps the enzyme catalyze the reaction more effectively. So to summarize, enzymes are essential biological catalysts, helping reactions proceed efficiently at body temperature. Their function is closely tied to their 3D structure and their dynamic way in which they interact with substrates. We'll discuss some factors that influence enzyme action. First, let's talk about temperature. As temperature increases, so does the kinetic energy of the molecules. This means enzymes and substrates move faster, which leads to more frequent successful collisions between them. As a result, the reaction rate increases as temperature rises. However, there's a limit. At around 50 degrees centigrade, the enzyme structure begins to break down due to excessive heat. This causes the enzyme's active site to change shape, a process known as denaturation. When an enzyme denatures, it can no longer bind to the substrate, effectively halting the reaction. It's important to note that each enzyme has an optimum temperature, a specific temperature at which it works best. For many enzymes in the human body, this is around 37 degrees Celsius, which corresponds to the normal body temperature. At this point, the enzyme operates most effectively, and any deviation from this temperature, either too high or too low, can in turn reduce its activity. So now let's move on to pH. pH is the concentration of hydrogen ions, or H+, in a solution, and this plays a key role in enzyme function. Changes in pH can affect the charge of amino acids in the enzyme, especially those in the active site. This can alter the enzyme shape, making it less effective, or even preventing the substrate from binding altogether. Just like temperature, enzymes have an optimum pH at which they work best, and deviation from this can disrupt their function as well. So to summarize, temperature and pH are both critical to enzyme action. Higher temperatures increase reaction rate up to a point, but beyond that, the enzyme risks denaturing. Similarly, changes in pH can alter the enzyme's active site, making it impossible for the substrate to bind. Understanding these factors helps us explain why enzymes function so effectively at specific conditions. Two more key factors that influence enzyme action. The first being enzyme concentration. Enzymes catalyze reactions by binding to substrate, so it makes more sense that the more enzyme molecules you have, the faster the reaction should go. This is true as long as there is an excess of substrate. In these cases, increasing the enzyme concentration will proportionally increase the reaction rate, as more enzymes are available to form enzyme-substrate complexes. However, if the substrate is limited, adding more enzyme will no longer speed up the reaction. This is because there simply aren't enough substrate molecules to occupy all the active sites, so the excess enzymes remain unused and useless. In this scenario, the reaction rate levels off because there's not enough substrate to match the increased enzyme concentration. So now let's consider substrate concentration. As you increase the substrate concentration, the enzymes work faster because more collisions are happening between enzyme molecules and substrate molecules. The reaction rate rises as more active sites are occupied, allowing for more enzyme substrate complexes to form. But this increase in reaction rate only continues up to a point. Once all the active sites are fully occupied, adding more substrate will no longer increase the reaction rate. The enzymes are working at maximum capacity, and no further enzyme substrate complexes can form because there are no free active sites left. This is known as the reaction reaching saturation. So to summarize, both enzyme and substrate concentrations have a significant impact on reaction rates. When there's plenty of substrate, increasing enzyme concentration speeds up the reaction, but when the substrate is limited, adding more enzymes doesn't help. Similarly, increasing substrate concentration boosts the reaction rate until all active sites are occupied, at which point the reaction rate plateaus. Finally, we're going to discuss enzyme inhibition, which reduces enzyme activity. Enzyme inhibitors are substances that reduce enzyme activity by either directly or indirectly interfering with the enzyme's active site, which is where the substrate binds. There are two main types of inhibitors, competitive and non-competitive, and each affects enzyme activity in different ways. Competitive inhibitors have a shape that is similar to the substrate, so they can compete for the active site of the enzyme. When a competitive inhibitor occupies the active site, it temporarily blocks the substrate from binding, slowing down the reaction. However, this type of inhibition is reversible. If you increase the substrate concentration, the substrate has a better chance of outcompeting the inhibitor, so the inhibitor's effect is reduced. Similarly, if you add more competitive inhibitor, it takes longer for the reaction to complete, but eventually it does, since the inhibitor is not permanently bound. On the other hand, we have non-competitive inhibitors, which work slightly differently. They don't compete for the active site. Instead, they bind to a different site on the enzyme, which in turn alters the shape of the enzyme, including its active site. As a result, the substrate can no longer bind to the active site, and the reaction is eventually halted. Unlike competitive inhibitors, increasing the substrate concentration won't reduce the effect of non-competitive inhibitors because the active site has been physically altered, making it unusable in the future. So to summarize, inhibitors are crucial in controlling enzyme activity. Competitive inhibitors temporarily block the active site and can be overcome by adding more substrate, while non-competitive inhibitors change the shape of the enzyme and cannot be outcompeted by substrate, effectively stopping the reaction altogether. Thank you much for taking the time to watch this video on enzymes. We discussed enzyme action, factors that influence enzymes and their rates, and enzyme inhibition. If you enjoyed, make sure to like, comment and subscribe and share to your friends. As well as that, feel free to check out any other videos on the channel, as there's many other videos on A-level biology discussing many other topics. Thank you.